Rules governing genetic code. The genetic code is subject to 3 rules that govern the arrangement and use of codons in the messenger RNA. 1. Codons are read in a 5' to 3' direction. 2. Codons are non-overlapping and the message contains no gaps. 3. The message is translated in a fixed reading frame which is set by the initiation codon. Reading frame is a series of nucleotides read in sets of 3 codons. Only one reading frame is correct for encoding the correct sequence of amino acids. Most amino acids have more than one codon. Only methionine and tryptophan have a single codon. Also, multiple codons corresponding to a single amino acid usually differ only by the third base. For example, GGU, GGC and GGG all code for glycine. This situation is called degeneracy, that is, the code is degenerate. Start codon. AUG codes for both methionine and the start of translation depending on the circumstances. In eukaryotes, the start codon codes for methionine, while in prokaryotes it codes for formilated methionine. There are also alternative start codons in both eukaryotes and prokaryotes. They still code for methionine and only the initiator tRNA is different. For example, CUG in eukaryotes, GUG, UUG, AUU and CUG in prokaryotes. Termination, stop or nonsense codons. 3 of the 64 codons UAA, UAG, UGA do not code for any amino acid. They are termination codons which, when one of them appear in an mRNA sequence, it indicates finishing of protein synthesis. They are also called stop codons dedicated to ending translation. The 3 stop codons also have colloquial names. UAA stands for ochre, UAG for amber, UGA for opal, with UAA being the most common in prokaryotic genes. Characters of the genetic code. First one, specificity. The genetic code is specific, that is, a specific codon always codes for the same amino acid. Second one is universality. The genetic code is universal, that is, the same codon is used in all living organisms, prokaryotic and eukaryotic. The third one is degeneracy. The genetic code is degenerate, that is, although each codon corresponds to a single amino acid, one amino acid may have more than one codon. For example, arginine has 6 different codons. Wobble hypothesis. There is a phenomenon called wobble in which a codon-anticodon match is allowed and stabilized for translation even if the nucleotide in the third position is not complementary. Many amino acids are specified by more than one codon. It is also known as redundancy. Frequently, a tRNA can translate more than one of these codons, sparing the cell from making multiple tRNAs to carry the same amino acid. For instance, the tRNA for arginine can translate both the CGA and CGG codons that specify arginine. Correct base pairing is required at the first position of the codon, that is, third of anticodon, and the second position of the codon, or second of anticodon. The third position of the codon does not always need to be paired with the anticodon. For example, it is allowed to wobble in some cases. In most instances, the third base is the degenerate base. It is sometimes referred to as the wobble base. In 1966, Francis Crick devised the wobble concept. It states that the base at the 5' end of the anticodon is not as spatially confined as the other two, allowing it to form hydrogen bonds with more than one bases located at the 3' end of a codon. It is not necessary that a specific tRNA anticodon would exist for every codon. Highly purified tRNA species of known sequence could recognize several different codons. An anticodon base was not one of the 4 regular ones, but a 5th base, inosine. The Wobble rules. 5' anticodon base G can pair with U or C at the 3' codon site. 5' anticodon base C can pair with G at the 3' codon site. 5' anticodon base A can pair with U at the 3' codon site. 5' anticodon base U can pair with A or G at the 3' codon site. 5' anticodon base I can pair with U, C or A at the 3' codon site. Gene mutation. Altering the nucleotide sequence leads to changes in the codons and thereby may even alter the amino acid sequence too. Changing a single nucleotide base on the mRNA can lead to any one of the following 3 results. The first one is silent mutation. The codon containing the changed base may code for the same amino acid. Second one is missense mutation. The codon containing the changed base may code for a different amino acid. The third one is nonsense mutation. The codon containing the changed base may become a termination codon. Decoding the genetic code. The decoding of the codons on the mRNA to a meaningful amino acid sequence is possible by the help of a tRNA molecule and a set of enzymes called amino-acyl tRNA synthetases. 3 regions of each tRNA molecule are used in the decoding of mRNA. They are anticodon, amino acid attachment site, and recognition region. The 3-dimensional structure of tRNA molecule was first determined through X-ray crystallographic studies by Alexander Rich and Aaron Klug in 1974. For each codon in the mRNA that specifies an amino acid, there is one tRNA molecule containing a complementary sequence of 3 adjacent bases that can pair with the codon on mRNA. The correct amino acid is attached to the other end of the tRNA. When it identifies the appropriate anticodons, it comes and attaches to the mRNA, which initiates the translation process.